moving on to number 16. 16 and plus. Number 17, I am marking here that 3 different parts of the urethra, which I was surprised if you didn't answer them on the test. You just repeated them before, just a few minutes before you take your exam last time. Urethra has 3 parts, if you remember. Part that travels in the prostate gland, and this was my prostatic urethra. Then we're going to have the membranous or intermediate urethra, and then the part that passes in the penis, we call it either spongy or penile urethra. Alright, so again, 3 parts here. So the main thing is called urethra. All of it together is urethra. And 3 different parts. We have part that passes in the prostate gland, is going to be prostatic. We have part that passes between the prostate and the penis. This is intermediate or membranous urethra. And we have the part that travels in the penis. This is non-spongy or penile urethra. Number 18 is the external opening of the urethra that would be releasing semen and the urine out. This is the external urethral orifice. What do you think? What do you think about this material? Is it easy or difficult?
It's challenging.
I don't think it's difficult, but I think it's just a waste of some of the questions, or it's hard to tell exactly which.
I think it's harder to identify with some of the how it's drawn, but
Yeah,
It's
because it
harder
looks different in
to
diagrams.
identify stuff on the drawing
right,
rather
specific,
than on the diagram.
right.
So if you got it on the diagram, you would be able to identify it.
Now, yeah.
So those are straightforward questions. You need to be able to create a pattern to study the diagrams with in order for them to make sense to you, rather than just trying to memorize where the arrow is going. This is the first thing that I got, that I did figure out from your test last time. That most of you are trying to have a photographic memory and memorize where am I pointing to, and this is, says in the textbook, this words. Alright, so you need to create a pattern. Yes.
I know you keep referring back to the lack of test. I think me personally, what threw me off was the usually you will have a word bank.
As I told you, I will not, or I will give you a word bank. You should be prepared as if you will not be getting a word bank.
Okay, I'm just saying that there's,
Yeah.
you're saying about it, but that
Don't
kind of threw me off.
expect me to give you word banks. And this is, I did make it on purpose to not give you a word bank for me to make sure that you should know how to spell the words. Because try to imagine that in 2 years you're gonna be the nurse and you won't be able to spell urinary bladder infection, for example, or peritoneal abscess, or operator obstruction. Whatever. So then you're gonna be writing the report in the hospital. So who did teach you? You can tell like me, right? So you should know the spelling, of course. Alright, so this is actually the information that represents 25% or more of the upcoming exam. So nothing that I will be creating new on the test. So we have the information. We just need to spend the time after the class. So how will I identify where is it from? If you didn't get it right, from your memory, because you did study, you're doing a good job in whatever number of questions you got right. Alright? This means that you did go and read the material after class, and this is what I highly recommend, because if you waited 2 to 3 days after class and you didn't read the material, through the material, as if you did not print the lecture, and you wasted 2 to 3 hours of your time. Alright? The number of questions that you have this time next to that. This means, yes, you did pay some attention in class, but you didn't read. You had a good memory from class, but you didn't go through the material on your own. You didn't put enough effort to study the material. This means if you did not pay attention in class and you did not review the material. So calculate what percent you have for each, and you should be able to figure out where you should be working more. Should I be paying more attention in class, or should I be reviewing at home, or I'm good to go? Alright. Questions? Was this stressful? Yes. Yes, this is what I want.
Yeah, it was stressful.
You activated, I need to put some stress on it. Alright? So, questions? Was this helpful?
Yeah.
Figure out
Yes, sir.
stuff?
Yes, sir.
So this is what I usually ask you to do by yourself, to test yourself, to know what you know and what you don't know. Alright, you want to figure out that you don't know until you test yourself. Very, very important. Alright? So again, if you remember from last time, we did 4 gametes. What is the one? Gametes is a lot of fun one. If you remember what makes meiosis different from mitosis, I have one cell division following one duplication of my genetic material. So if I did duplicate the genetic material, I have now 4N. You're gonna perform one division, so you're gonna be forming 2 identical daughter cells, which will resemble the mother cell. So 1 cell division after 1 replication of DNA. This is mitotic division. Meiosis means I will be performing 2 successive cell divisions after 1 duplication. So you're going to be dividing the cell like this, 1 time, and then another time. So what you will end up with, you will end up with 4 cells, and each one of those 4 cells has only half of the genetic material. And why do I carry only half of the genetic material? For me to use those gametes in fertilization. So you are fusing 2 gametes, either an egg with a sperm, to form a new cell that has a combination of both your genes and your partner's genes. Alright? How will we produce sperms? This process of the sperm production, this is called spermatogenesis. How do I form sperms? The process of the formation of sperms is called spermatogenesis. Spermatogenesis has 3 phases. First phase, I will use my stem cells for the sperms. Those are the spermatogonia. To perform a mitotic division, I will be forming 2 daughter cells which will have a complete set of information. Those are my primary spermatocytes. So again, you start with the spermatogonia. This is the stem cell for sperm production. Start with the spermatogonia. Has a complete set of the genetic material. You divide it by a mitotic division, mitosis. Can you remind me, mitosis is going to be how many cell divisions? One cell division after how many replication of the DNA? 1. So what's this will result in? 2 daughter cells, which will have a complete set of the genetic material. Those cells that I will be producing, those are my primary spermatocytes. This is my primary sites. Primary spermatocytes. Primary spermatocyte will then divide by meiotic division. I will add meiosis on here. So what happens to this site, spermatocyte, I will divide into 4 cells. Each cell has only half of the genetic material. What do we call the cells on here? Those are my spermatids. So again, what is the first step in spermatogenesis? You divide the cell by mitosis. And then you're going to be dividing the primary site by meiosis. This will result in information on how many haploid cells. 4 haploid cells. And those 4 haploid cells are going to be called my spermatids. Are those sperms? What do you think? Yes or no? They are not sperms. I need to have a modification in the shape of those cells. I have the same genetic material like a sperm, but those cells cannot travel for long distances. I need to modify the shape of those cells. I will be passing here through spermiogenesis. Spermiogenesis is change in the shape of the cell. So I will be forming the final shape of the sperm. And you see the sperm will have head, middle part, and tail. I will be accumulating digestive enzymes at the apex of the head of the sperm. Behind it, we're going to have the nucleus. And in the middle portion here, I will carry lots of mitochondria to provide the sperm with energy for it to keep swimming through the fluids. All right? So the process here of the shape modification, I did not perform any cell division on here to transform spermatid into a sperm. It's just a modification of the shape of the cell. This process is called spermiogenesis. spermiogenesis. So again, the whole process of the sperm production, this is called spermatogenesis. Don't confuse spermatogenesis with spermiogenesis. Spermatogenesis means you are performing mitosis, then meiosis, then spermiogenesis. What is spermiogenesis? This is a modification of the shape of the spermatid into a sperm. Mitotic division is what prevents, is the reason why males won't have menopause. Why men won't have menopause? They, theoretically speaking, will be fertile for life. Why? Because I am producing the same cell. I am performing a mitotic division, so I have an unlimited number of sperms that I can produce. This mitotic division will form two daughter cells which are identical and look the same like the mother cell. In females, as you're going to see after break, mitosis will stop after birth. So females will have a limited number of primary eggs. So I will be consuming them over a period of time, and that's it. But mitotic division in males will be taking place for life. That's why males won't have menopause compared to females will have menopause. What I mean by menopause, it's my ability to produce gametes will stop. If I'm not able to produce eggs anymore, I have menopause. Why we don't have menopause in men? I can't produce sperms for life simply because of this mitotic division. I'm able to duplicate the stem cells, the spermatogonia. All right? Mitotic division is taking place all the time. All right? So what is the difference between the gonia and the primary site? If it keeps dividing, I will call it primary site. If it turns into another stem cell and it did perform another mitotic division, I will call it a gonia again. This is the same cell but with two different names. Does this make sense? Yes. If I keep going down, I will call it primary spermatocyte. If I will be performing another mitotic division, it's going to be a gonia, and so on. All right? Questions? Yes.
All right. So what is the arrow pointing to? Yeah.
What arrow?
In the sperm. Yeah. Right there.
Those are the digestive enzymes that will be breaking down the mucus and will be able to penetrate through the egg. It allows the sperm to penetrate through the corona mediata around the egg. Here is the nucleus, and here I have the mitochondria that will be producing the ATP needed for the sperm to be activated. Right?
Sorry. And could you repeat what's the difference between spermatogenesis and
Spermatogenesis is all the process. So the transformation of a gonia to primary site by mitosis, and then the transformation of the site into a spermatid by meiosis, and then the transformation of the spermatid into a sperm by spermiogenesis. All this together is spermatogenesis. The change in the shape of the spermatids into sperms, this is a modification of shape. I did not perform any division here. I did not change the DNA. I did not change the number of chromosomes. I'm just changing the shape of the cell. This is called spermiogenesis, the transformation of a spermatid into a sperm. Questions? So again, what we can see on here, we have spermatogonia. Spermatogonia will divide by mitotic division. I will form primary spermatocytes. Primary spermatocytes will divide one division, meiosis 1. I will become a secondary spermatocyte. Then I will divide another division, meiosis 2. I will form my spermatids. Are spermatids diploid or haploid cells? Haploid cells. They have half of the genetic material. Do I need to perform any other cell divisions? No. You just need to modify the shape. So this oval shaped or circular shaped cell will not be able to travel. So I need to have a tail that has the ability to move. I need to have a site with mitochondria. I have to have an apex with digestive enzyme. I need to make a conformational change of the cell for it to become a sperm that is able to allow fertilization to take place. Yes, you have the same genetic material, but you are changing the shape. What do we call this process again? Spermiogenesis. Spermiogenesis. A very important cell that we need to talk about when we describe spermatogenesis is going to be Sertoli cells, or another name for the Sertoli cells is going to be the sustentator cells. So we have two names for the cell, same cells. Those are either Sertoli cells or sustentator cells. Sustentator cells or Sertoli cells, those are like the nursing cells for the stem cells of the sperms. They will be providing nutrients, they will be providing the signals needed for spermatogenesis to take place. Any excess cytoplasm that will be released, that will be sloughing off during the spermiogenesis, during the transformation of the spermatid into a sperm, this extra cytoplasm that you're going to be getting rid of, this will be ingested and taken away by your Sertoli cells. Also, Sertoli cells will be releasing the fluid through which the sperm is going to be traveling in the seminiferous tubules. See up here. Before I get the secretions from the seminal vesicle, how the sperms will travel through the seminiferous tubules, the weak testes, epididymis, vas deferens, all this path, how would I be able to travel? I am traveling in the fluid secreted by the sustentator or Sertoli cells. So sperm need a medium to swim inside. Alright, so what will be releasing the fluid that will be conducting those sperms all this way until I get the secretions from the seminal vesicle is going to be my Sertoli cells. Alright, so again, what are the three main functions of Sertoli or sustentator cells? I am providing the nutrients to the stem cells of my sperms. I will be disposing the excess cytoplasm that will be sloughing off during spermiogenesis and providing the signals needed for the cell division. Also, I will be secreting the fluid that the sperms will be traveling in until it reaches the seminal vesicle. All right? Does this make sense? Questions? All right, so according to what you're going to be producing sperms, what would tell your testis you need to produce sperms, or you've got enough, don't produce anymore? What do you think? What controls spermatogenesis? Hormones. brain that will be located below the thalamus. In the brain, this is called the hypothalamus. And the hypothalamus is going to be responsible to release hormones secreted from the posterior pituitary gland. Also, it's going to be responsible to regulate the hormones released from the anterior pituitary gland, if you remember. Anterior pituitary gland is going to be releasing two hormones that we're concerned with. Those are my follicle stimulating hormone, FSH, and luteinizing hormone, LH. What stimulates the anterior pituitary gland to release FSH and LH is the hypothalamus. Hypothalamus is going to be releasing a hormone called GnRH, or gonadotropin releasing hormone. So again, hypothalamus is going to be stimulating the anterior pituitary gland to release the gonadotropins, and the hormone that will be released from the hypothalamus to stimulate the anterior pituitary gland to release the gonadotropins is my gonadotropin releasing hormone. It is a hormone that causes the release of the gonadotropin. And from its name, from the name, what am I activating? The gonadotropin. What am I activating? Tropin is to activate. So if I'm called gonadotropins, I am activating the what? Gonads. So in males, what are the gonads? The testes. Each one of those two hormones is going to be affecting a different type of cells in the gonads. FSH and LH. LH is going to be affecting my interstitial cells. And the interstitial cells is going to be responsible to release the androgens, male sex hormones. And the circulatory cells are going to be affected by FSH, follicle stimulating hormone, to release androgen binding protein. And the androgen binding protein, or AP2, is going to be acting as a surface receptor on the oval of the spermatogonia for the androgen. So again, how do I regulate spermatogenesis? First, the hypothalamus releases GnRH, gonadotropin releasing hormone, that will stimulate the anterior pituitary gland. Anterior pituitary gland, once it gets stimulated, it's going to be releasing FSH and LH. FSH is going to be stimulating what cells? My Sertoli cell. Sertoli cell is going to be releasing an androgen binding protein. LH, released from the anterior pituitary gland, is going to be stimulating another type of cells in the testes. This is to increase the muscle bulk. Testosterone. Injections. What he is trying to do, he is trying to get secondary boost of the secondary sexual characteristics. And one of the secondary sexual characteristics is increase in the muscle bulk. So what's going to happen at this point? If I have lots of androgens, how this might cause infertility?
It will shut off the natural reproduction
Of the androgen, right?
of the testosterone.
Of the testosterone, yes, but and testosterone is a type of androgen. Right, so we are
relying on the injections instead of the natural
So, but why would you need natural if you could have external? Why is the external did not act on the spermatogonia and enhancing those cells to divide? Actually, he should be producing much more sperms than normal.
Because he doesn't have enough
Binding protein. So if I am having lots of external androgens, what's going to happen? This will have a negative feedback on my pituitary gland and on my hypothalamus. So I'm not releasing GnRH, I'm not releasing FSH and LH. And if I'm not producing FSH, would I be producing the androgen binding protein?
No.
No. And if I don't have the androgen binding protein, will there any benefit to have lots of androgen in terms of spermatogenesis? No. Because simply, yes, I have androgens, but I don't have the receptor for those androgens on the surface of my cells for me to transduce the signals to the inside to enhance the cell division. So yes, I have lots of androgens, but those androgens will not have any effect on my cells simply because I don't produce those receptors anymore. Because of the negative inhibition, negative feedback on my hypothalamus. So if you notice on here, GnRH stimulates anterior pituitary gland, FSH, LH will be produced. FSH acts on what cells again? My Sertoli cells. Sustentacular cells is another name for it. Compared to my LH, it's going to have an action on what cells? My interstitial cells. Sertoli cells will be releasing the signal here. This is my androgen binding protein. And the interstitial cells are going to be producing the testosterone or the androgens in general. This androgen will bind to the androgen binding protein, and this what's going to stimulate my spermatogenesis to take place. Division of the gonia into primary sites, into secondary sites, into spermatids, and then modification of the shape of the spermatid into a sperm. All right? Questions?
And FSH was it? You said follicle what?
Follicle stimulating hormone. Follicle stimulating hormone, and we're gonna see after the break why did we call it follicle stimulating hormone? Because it was first discovered in females, not in males, and it stimulates the follicle, as we can see. All right. So again, hypothalamus releases the gonadotropin releasing hormone, GnRH. GnRH can stimulate my anterior pituitary gland to release FSH and LH. FSH will act on my sustentacular cells or my Sertoli cells. This will enhance the cells to release the androgen binding protein, which will act as receptors for the male sex hormones on the spermatogonia. LH will stimulate the interstitial cells to release my androgens. Alright, again, you need androgen and the androgen binding protein for it to have an effect on your spermatogonia. Alright? So what do we call the nurse cells that will help in the development of sperm? Sertoli. Cells, other name for it is sustentacular cells. Is there any other effect of the androgens on the body other than the formation of sperms? Yes, the appearance of the male secondary sexual characteristics. Like what? Like appearance of pubic and facial hair, deepening of the voice, thickening of the skin and make it oily, bone growth, and enhance the density of the bones, also increase the muscle size and bulk. All those are secondary male sexual characteristics due to the presence of the male sex hormones. So male sex hormones, primary function, primary sexual characteristic is the ability to produce sperms. Other stuff are the secondary sexual characteristics. All right? Questions?